Okay, to conclude, right? To conclude, basically, again, you have to identify the fragment that you want. All right. How do you identify it? You can. We have, of course, we haven't looked at PCR and some other specific methods to amplify DNA, but basically, you have to identify the fragment of interest. You know vectors. You have to choose the suitable vector. In this case, all right, we'll just use a plasmid. You cut the plasmid and the fragment so that with enzymes so that they can match, right? If you cut them with blunt ends, then it's not easy to do. You just random. They can you can still cut, you can still join them, but the efficiency is very low. But if you cut both ends with matching protruding ends, so that means that's why it's called sticky ends. It'll increase the chances of ligation much higher. So you cut with resistant enzymes, you put them together, and then now you want to seal it. You seal this match. You seal it with the ligase, all right? So then, what you can do then is you put this back into the plasmid, and this is a process called transformation. Bacteria does that. Bacteria likes to do that. It likes to take in. This is how most bacteria eat. They travel, and then if they find something, they'll just envelope the whatever compound and take it in. And if it doesn't need it, it will take it out. All right. This is the same. This is the same way you exploit this bacterial property to take in your pieces of DNA. But you can help the process by treating the bacteria with, say, for example, a heat shock process. There are some chemicals you put in, and then what you do is you actually make pores on the bacterial wall so that it's easier for your DNA to get in. You injure the bacteria. But then again, after the process of transformation, then you let the bacteria revive, repair itself, then before you let it make copies again. All right, this is where you need selection. Okay, because when you mix your plasmids with your when you want to you insert your plasmid inside your host cells, you'll just mix them again in a tube. There will be some bacteria that doesn't take your plasmid. There will be some bacteria that does take your plasmid, and so on. So you need to be able to choose, and this is where selection is important. You normally grow this in the presence of antibiotics. You make sure that your plasmid contains an antibiotic resistance gene. So so if it has antibiotic resistance ampicillin, so now you grow the bacteria in the presence of ampicillin so that all other bacteria will die. Only bacteria that has your plasmid will be able to grow. All right. Once you have that, then you regrow them. So you make you bulk. You grow them in bulk because you want a lot more, right? And then depending on what you want to do, if you want just to make copies of it, then you can get the DNA back out, or you want the bacteria to produce the proteins. You put in some of the things that you consider is that if you want to make copies, you can just put whatever fragment. If you want to make proteins, so you make sure that the fragment that you put in are complete genes, like from start to end, so that transcription can occur. You need to put a promoter in. You have to put the whole complete set of what defines a gene to be expressed inside E. coli or whatever host. So once you be able to do that, and then you'll be able to extract the protein of interest. This one just shows you how another way of selection. Remember I mentioned that we can select based on presence of antibiotics. So here, because sometimes, this one gives you a double selection. Why? Because there are two processes that you want to select. The first is that you have your fragment and you have your plasmid, right? You cut them with enzymes, you join them together. How do you join them? You put them in a tube, your vector, your plasmid put in, and then you put in your enzymes so that hopefully I say hopefully because there's no way for you to control it, but what you want is actually your fragment and your plasmid to create a recombinant DNA. But what can also happen is that your plasmid can just join back together and doesn't have your insert, right? Now, this slide will be able to allow, it allows you to select which plasmid contains your fragment, okay? You clone, why, see, look at this white region is called the LECZ gene. What does LECZ do? You know, it breaks down lactose to by beta galactosidase and so on. Correct. Now we exploit that system where um, you use the beta galactosidase to actually interact with a different a gene, the XGAL, and then you put in the IPTG. You know, these are, you can read them if you want, but basically what it does is that if you have XGAL product, the beta galactosidase, if it is active, that means you'll get blue color. If you clone your fragment inside that LECZ, because you see, the registration site is actually inside the white fragment. So if you put your gene inside there, what you are also effectively doing is you are breaking the white gene, correct? So the white gene no longer functions, correct? This LECZ gene. And when you clone this, you get two types of colonies. White colonies and blue colonies. Why do you get two types? Remember, I mentioned if the LECZ gene is still intact, it will produce the beta galactosidase and so on, and then it will react with the system, the IPTG, and so on. It gives you a color blue. But because you have inserted your fragment within the LECZ gene no longer functions. And because of that, your colonies becomes white. So, when you have this colony, the blue and white, which one do you want? You want the blue or you want the white? If you take the blue, you're going to have the plasmid, but you don't have your fragment of interest. What you actually want is the white, because this are plasmid that contains your fragment of interest. So this is another, and then you can use this also to grow your bacteria in the presence of antibiotics. Right? So it's why I say it's double selection. One, you select for bacteria that has your fragment, and it also allows the bacteria that carries the plasmid and your fragment to grow using antibiotic resistance. All right. Okay. So I hope that gives you a sort of an overview of what cloning is about. Again, this is not meant to be a full-fledged course on recombinant DNA technology. It just means to give you a very brief introduction on the processes, and so this is what I'm trying to do. So if you have any questions, as usual, if you have any questions, please let me know, and then I will explain a little bit more if you need me to. But having said that though, so this is the last of the series of videos for molecular genetics. So I wish you all the best, and I'll see you in other lectures, other courses in the future. All right, bye-bye.